Okay, so hello everyone. So today I'll be talking about the application of biological large language models for AI-driven drug discovery. And it starts early. Okay, cool. Just takes a small delay. And before getting into, I guess, the more exciting part, which are the BioLLMs, I want to give you a brief overview of drug discovery and development. I'll make this quick because my background is in machine learning, not drug discovery. So the last time I spent here, the last wrong things I'll say. Yeah, and so basically the drug discovery and development life cycle is a very expensive and very time-consuming multi-step process. So you can think of it as a funnel, right, where you have a bunch of initial possible compounds, drugs that may target your disease, and then you go along these funnels, filtering them down across more and more strict filters to eventually get to the drug that you actually will cure your disease. The phase where we'll be focusing on today is stage 1, so drug discovery, typically because this is where AI and machine learning is mostly applied on. And there are 3 main phases in this stage. So there's target identification, lead identification, and lead optimization. Before zooming in there, let's just get out of the way what are targets and what are leads. So targets are typically proteins. They can also be DNA, RNA, but for today we'll be focusing on proteins. And basically proteins are what do stuff in your body, right? And these target proteins are the ones that you want to inhibit to cause some downstream effect on the disease, right? So basically they are the targets that if you inhibit, hopefully will help you cure disease. Then you have compounds that are the leads, which are typically small molecules, so molecules small enough that they can diffuse across cell membranes and then interact hopefully with targets. And basically they will bind to these targets to inhibit their function. And so many of you, maybe not knowingly, have consumed small molecules. So if you've taken ibuprofen or remdesivir, you've been taking small molecules. So now let's look at the actual drug discovery funnel and see how targets and leads interact. So the first step, as I mentioned before, is target identification. This is a funnel where you start by all of the possible proteins that you can target to cure the disease. And intuitively, you can think about it like this. So maybe you can grab a population that has the disease, another population that does not have the disease, and then you see, for example, in the population with the disease, if there's a certain type of mutation that only happens there. So that will give you maybe some indication that those mutations could be, those mutated proteins could be the targets for your drug. Then there are also other types of approaches, right? Because this is more of a correlation-based methodology. There's no intervention, but there are more causality-based approaches like RNA interference or CRISPR, which you may have heard about. But TLDR is at the end of this phase, you want to have a short list of target proteins for your drug. Then the next phase is lead identification. So finding the small molecules that will bind to your target. So here you typically start from the universal molecular diversity, which 10 to the 63, so pretty much infinity. So here the challenge is how can you find the actual molecules that will bind to your target. So this is again a funnel, right, where there's going to be mostly lab experiments where you're going to test many of these small molecules. First filtering them based on domain knowledge to get to a short list of small molecules that actually are capable of binding to your target. And then finally, you have the last phase, which is lead optimization. And basically, this phase is important because having a small molecule, a lead that binds to your target is not enough, right? You also need it to have other properties, drug-like properties like having low toxicity, having high solubility. And so basically, you need to balance efficacy and safety. And so as you can see here, the drug discovery is a lot about these funnels, right, where you're going along the pipeline and filtering down until you eventually get to a good short list of small molecules which are good candidates for your drug. But the thing is that all of these funnels involve a lot of lab experimentation, which is expensive and time-consuming. And so where AI and machine learning come in is as a way to allow us to scale these funnels, right? So the whole idea about computational methods like AI, machine learning, but also simulation, is that you can cover the initial part of these funnels doing virtual experimentation. So that's typically called in silico. So here the idea is that you're doing all of these experimentations in the silicon of the computers, right? Not in the lab. And the whole idea here is that this type of screening, this type of experiments are much more scalable. And so this is where basically AI and machine learning really come in the drug discovery funnel, is to help us screen and experiment all of these virtually, and then optimizing and minimizing the amount of compounds that we actually need to then test in the lab. Okay, so hopefully now you're thinking, that kind of makes sense, but where do the BioLLMs actually come in? And so if you think about it, the sequences that we've been talking about, so proteins, molecules, DNA, which I didn't really talk a lot about, they are all sequences of symbols that belong to a known vocabulary, right? So proteins are made up of amino acids, and molecules are made up of atoms and bonds, and DNA of nucleotides. And so if you think about them, they are basically languages, biological languages. And so here, a second, trying just to pass the slide. This is a good thumb exercise. Okay, but here the key premise is that just as LLMs, just as LLMs have transformed how machine learning is applied to natural language, BioLLMs, biological large language models, are transforming how machine learning is applied to biological languages. So maybe let's take a step back here and just think a bit about what are LLMs and BioLLMs overall, which at the end of the day, they're like pretty much the same thing behind the scenes. So I like to think of them as having 3 key ingredients. So one of them is vast amounts of unlabeled data. So typically for a natural language, this is like the whole internet. And for BioLLMs, it's like databases of protein sequences, DNA sequences, molecules, all of that. And actually, like this type of biological data has been growing pretty much exponentially for the last years with advances in high throughput screening, higher resolution technology. So we definitely have a lot of unlabeled data. Then there's self-supervised learning, which is this simple yet beautiful idea of how you can learn, train a model using unlabeled data. And I can put it better than LeCun, which basically said that here the idea is to predict, pretend that there is a part of the input that you don't know and predict it, right? So that's just it. Then more concretely, maybe you can do mask language modeling, where you mask some of the tokens in the sequence and then predict it, or more like next token prediction. But it's always the same thing. Let's turn the problem around so that we can actually learn from unlabeled data. And yeah, somehow that really gets us really good embeddings. And then the third ingredient is transformers, right? So sort of the backbone to modern LLMs, this neural network architecture, which just has been shown to scale really well to bigger amounts of data and to capture long-range dependencies. And so let me show you a specific BioLLM. So this is ESM2, which is somewhat popular, but maybe I'm in a bubble. And basically, ESM stands for Evolutionary Scale Modeling. It's a model developed by Meta, by Facebook. And basically, it's a protein language model. So basically, the data that they use to train it are protein sequences. They come from UniRef, and they used around 65 million sequences. Then, in terms of masked language modeling, so they did the tokenization per amino acid. So each amino acid, each character is going to be one token. They did a 15% masking rate, which is quite common in like BERT-like models. And if you look there at the image, basically that's what the tokenized sequence looks like. So the protein is each unique amino acid is going to get converted into a unique number, a token, and then you're going to mask some of those tokens, and that's what you're going to try to predict during training. And then finally, in terms of the architecture, it uses a BERT-like encoder. I think the main difference there is just that it uses rotary positional embeddings, ROPE. And then they actually trained multiple models, like multiple variants. So starting from 8 million, which is pretty small. I don't think anyone's ever used it besides for the paper. And then the 15 billion version. Another model that I want to talk about today is MolFormer, which is basically a language model for molecules. And so here they used, for example, Zinc and PubChem, which are databases of molecular data. And they trained in around 1.1 billion sequences using the SMILES form. Like these molecules were formatted in the SMILES format, which is just one particular way to turn into strings these bonds and these atoms. Then mass language modeling, pretty much the same thing. So tokenization is done per character, also a 15% masking rate. And then the actual architecture is also like a BERT-like encoder. But here they also used linear attention, so sort of an approximation of the quadratic attention, I guess just for make it a bit cheaper. And then they only released one model, which was the biggest that they trained, which is 46.8 million parameters. And so these are just two models that I talked about, but there's a whole landscape of models out there, the same way that we see with LLMs. And so this is basically a curated list of some of those models, and you can see it basically in a timeline, right? In the x-axis, the date that they were released, and in the y-axis, the number of parameters that they have, growing by 10 on each bar. And you can also see by the colors the type of language that was used. Is it DNA? Is it proteins? Is it molecules? And I think that there's a couple of things that pop out, some interesting insights that we can extract here. So one of them is, as expected, we can see that the number of parameters has been growing, as we see with LLMs. Then it's also interesting to see that proteins seem to be like the hottest language, right? That there's more protein language models than DNA or molecules. And then it's also interesting to see that there aren't really like big molecule language models. So like they all are more or less below 100 million parameters. Okay, so now I think we somewhat understand like the drug discovery and development pipeline, drug discovery life cycle, BioLLMs. So let's look into how we can actually merge the two, right? How can we use BioLLMs for drug discovery? So the first use case that I have here is simple. Maybe it takes a bit to get your head around it, but I think it's a really cool one. So basically, variant effect prediction is about estimating how a mutation in a protein, right? So you grab a protein, you change one amino acid, or you change multiple of them, how that mutation is going to impact the pathogenicity, so how pathogenic the function of that protein is because of that mutation. Okay? And I think it's somewhat intuitive that this is helpful, right? Especially in the target identification phase, like the first one, because probably if you're trying to identify targets for your drug, probably you want to focus on targets that actually, on mutations that are pathogenic, right? The ones that don't make a difference probably are not very important. And so the cool thing is that ESM, zero shot, can perform VP. So without any fine-tuning, without any labels, without much work, you can do VP with ESM. So how does that work? So you're going to grab your protein, right? Your wild-type protein, the initial one. You're going to mask the position that's going to suffer the mutation. You're going to run that through ESM. And ESM, as any BioLLM or any LLM, actually, what they actually do behind the scenes is they're going to predict the likelihood of all of the tokens that are in the vocabulary of being in that position. Right? That's basically what they are trying to do, to predict the likelihoods of these possible tokens, of these possible amino acids of being in that position. And so what you can then do with those likelihoods is calculate a score. And so this score is basically going to take into account the likelihood of the original amino acid, and it's also going to take into account the likelihood of the mutation. And what is really interesting is that if you see this score that is calculated, which basically means what is the likelihood of this mutation happening, right? With the vast amount of unlabeled data that ESM saw, what is the likelihood of that amino acid in that sequence being that one. That's really highly correlated with how pathogenic that mutation is. And it basically happens because pathogenic mutations are less conserved in nature. And so predicting how likelihood a mutation is, ends up being very inversely correlated with how pathogenic it is. And so grabbing ESM, zero shot, can really already help us solve a very useful task. Then let's get to a slightly more complex one. So this is DTI, drug target interaction prediction. And this is basically all that we do on the lead identification phase, right, on the second one. And so basically, what we want to predict here is the binding between the target and the leads, right, between the proteins and the small molecules. And this is something that is measured experimentally, right? This is something that we can collect. The thing is that it's very expensive to collect this, so as an ML engineer, let's use some of those labels, let's train a model, and let's predict it. And so that's basically what we are doing here. And how BioLLMs come in is that you're going to train a classifier model where you're going to represent the proteins and the small molecules using BioLLMs, right, using their embeddings. So you're going to grab your protein, you run it through ESM to get the embeddings that it learned for that protein. You're going to grab the small molecules, run them through MolFormer, and then you're just going to add some layers on top of that for doing your actual classification. Then you grab your labels, you do backprop, and then you update the weights of your classification neural network, right? Then concretely how you do this can vary a bit, depending on the trade-offs that you have. So maybe you can do pre-calculated embeddings, which is then going to put a bit of pressure on your data pipelines, like moving those embeddings around. Or you can do actual fine-tuning of the models, which is going to put a bit more pressure on the GPUs. But yeah, basically we've found that these pre-trained embeddings are very useful to capture these representations of proteins and molecules. And then finally, the last example that I have here, unlike the previous two, this is a generative one, right? The two previous ones were more about predicting, and this one is actually about generating something. And so this is for the lead optimization phase, which, as you might recall, is when we are like a multi-parameter optimization. We want to optimize multiple properties. And what we have here at the input is a molecule, right? This is a molecule that went through lead identification. It seems like a pretty promising molecule that it would bind to the target, but we want to make sure that it also has drug-like properties. And so there are some really cools out there like Reinvent4 and AceGen, which basically implement these reinforcement learning based workflows, where the whole idea is to use a generative model, so like a decoder like GPT, trained with small molecule data, and that's what's going to be generating new designs for your molecule. But the cool thing is that you're going to use this reinforcement learning workflow. So you're going to use scoring functions like, for example, the previous model that we just developed, the one that predicts the binding with the target. That could be one of the scoring functions. Then you're going to have other scoring functions like toxicity estimations and solubility estimations, all of those. And basically, all of these are going to provide rewards in the reinforcement learning loop to nudge the weights of the generative model in such a way that, as it is updated, it's going to generate molecules that are more and more aligned with your desired properties. And so basically, we've seen how AI and machine learning, and more specifically BioLLMs, can be applied along the multiple phases of drug discovery. And yeah, that's it for today. Hopefully, I got you to think a bit about how language models can be applied to more exotic languages, like proteins, DNA, and DNA, RNA, molecules, all of those. And yeah, curious to see if there are any questions.